Welcome to this deep dive. We're going to be looking at some really cool new research about cats.
Yeah.
And a possible genetic link to FSE susceptibility.
Exactly.
It's kind of a mystery.
Yeah.
Right now, we know cats can get feline spongiform encephalopathy, or FSE.
Right.
But we don't really know why.
Yeah. And this research might give us some clues.
Okay, cool.
This paper just came out like November.
Oh, wow. Hot off the presses.
Yeah, hot off the presses.
Okay.
It takes a real close look at the PRND gene in cats.
Okay.
And how like variations in that gene might be connected to FSE.
So we're talking about a gene, PRND, and how differences in that gene from cat to cat could potentially make some cats more vulnerable to FSE than others.
Exactly. And what makes it even more interesting is that the PRND gene is closely related to another gene.
Okay.
PRNP that we already know is involved in prion diseases in other species.
Before we get too deep into the PRND gene, can you remind us what prion diseases are?
Sure.
And why they're such a big deal?
Prion diseases are a group of fatal neurodegenerative diseases that are caused by misfolded proteins.
Okay.
And these misfolded proteins are called prions, and they can actually trigger other normal proteins to misfold as well.
Oh, wow.
Creating like a chain reaction of damage in the brain and the nervous system.
So it's not just that you have these abnormal proteins, but it's that they can spread and cause more and more damage over time.
That's exactly right. And that's what makes these diseases so devastating.
We know prion diseases exist in other animals besides cats, right?
Yes, we see it in cows with mad cow disease, in sheep with scrapie, and even in humans with Creutzfeldt-Jakob disease.
Yeah.
And the fact that FSC emerged in cats around the same time as the mad cow disease outbreak is particularly noteworthy.
Yeah, what's interesting about that timing is that while other species didn't seem to get FSE from consuming contaminated meat products, cats did.
Precisely.
Yeah.
That raises the question, why are cats seemingly more susceptible?
That's the question these researchers set out to answer.
Yeah.
And they focused specifically on the PRND gene.
Yeah.
Why PRND?
Well, PRND produces a protein that has some similarities to the prion protein.
Okay.
And researchers have found that variations in this gene are linked to prion disease susceptibility in other species, including humans and cattle.
Right.
So they wanted to see if the same might be true for cats.
So they basically thought if variations in this gene are associated with prion diseases in other animals, let's see if it's the same story in cats.
Yes. And to do this, they analyzed the PRND gene in 210 domestic cats.
Okay.
And what they found was remarkable.
Okay.
13 brand new variations.
Wow, 13. That's a lot of variation in just one gene.
It is.
What exactly are these variations?
These are what we call single nucleotide polymorphisms, or SNPs.
Yeah.
Think of them as single letter changes in the DNA sequence.
So like typos in the genetic code?
Exactly. And even a single letter change can have a big impact.
Of these 13 variations, are they all significant?
So out of the 13, they found 6 actually changed the amino acid sequence of the protein that the PRND gene codes for. These are the ones that really grabbed their attention.
So we're talking about changes to the building blocks of the protein itself.
Yes.
Which could potentially alter how the protein functions.
Exactly. In addition to these SNPs, they also found an indel that's short for insertion deletion.
Okay.
It's basically a chunk of DNA that's either missing or inserted where it shouldn't be.
So it's like a bigger typo in the genetic code.
Yes, a larger change that could also have significant implications for how the gene functions.
So now that we know what they were looking for and what they found, how did they figure out if any of these variations might be linked to a cat's susceptibility to FSE?
They use sophisticated computer programs to analyze the potential impact of these variations, particularly those 6 that change the amino acid sequence of the protein.
So they basically plugged these variations into a computer program and asked, could these changes cause problems?
Exactly. And out of the 6 they found, 4 were flagged as potentially problematic.
Okay.
These 4 are what we call non-synonymous SNPs.
Okay.
Meaning they change the amino acid sequence of the protein.
Okay, so now we have 4 variations that are particularly interesting.
Right.
Because they might actually disrupt the way this protein functions.
Right. One in particular called C.97A, greater than G, really stood out.
Okay.
Analysis suggests it might actually cause structural clashes within the protein.
Oh, well.
Potentially altering its shape and how it interacts with other molecules.
So this variation could be a real game changer in terms of how the protein behaves.
It could, and this is where it gets even more interesting.
Okay.
Because the researchers didn't just look at the PRND gene in isolation.
Okay.
They also wanted to see if there was any connection between variations in PRND and variations in PrPSc.
Okay.
That other gene we know is involved in prion diseases.
So they were looking for a link between our potentially problematic PRND variations
Exactly.
and variations in this gene, PRNP, that we know is linked to prion diseases.
Exactly. And what they found was a strong linkage between 2 of those potentially problematic PRND variations and an indel in the PRNP gene.
Okay, now we're talking 2 of the variations in the PRND gene that might be causing trouble are connected to a variation in the PRNP gene.
Right.
That we know is involved in prion diseases. What does that tell us?
That's the burning question.
Okay.
And that's where we'll pick up in part 2 of our deep dive.
All right, sounds good. Welcome back to our deep dive into feline PRND and its potential role in FSE.
Yeah.
We left off with this fascinating finding. 2 of the potentially troublesome variations in the PRND gene are linked to a variation in the PRNP gene.
Right.
Which we already know is involved in prion diseases in other animals.
Yes, and to remind everyone, this particular variation in PRNP, the indel is thought to be linked to how quickly a prion disease progresses.
So if a cat has this particular indel in its PRNP gene, the prion disease might progress more rapidly.
That's what researchers believe based on observations in other species.
So back to this link, why is it so important that these 2 potentially problematic PRND variations are connected to this indel in the PRNP gene?
Well, it strengthens the idea that the PRND gene might play a more significant role in FSE susceptibility than we initially thought.
Okay.
It's almost as if we thought PRNP was acting alone, but now it looks like PRND might be involved as well.
So maybe PRND is an accomplice.
That's one way to think about it. This finding suggests that variations in PRND could be indirectly influencing how susceptible cats are to FSE.
Okay.
And potentially even how quickly the disease progresses.
This is really giving us a new perspective on FSE in cats.
It is. Remember, this is the very first study to investigate PRND in cats.
Right.
So these findings are truly groundbreaking.
Yeah.
Identifying these new genetic variations and their potential connection to FS susceptibility is a significant step forward in understanding this disease.
It sounds like we've learned a lot about the role of PRND, but let's go back to the initial question that started this whole investigation.
Okay.
Why do cats seem to be more susceptible to prion diseases than other species? Does this research provide any insights into that?
It does. One thing that really stood out to the researchers was the sheer amount of variation they found in the feline PRND gene.
Okay.
This level of variation is similar to what's been observed in other species known to be susceptible to prion diseases, such as humans, cattle, and sheep.
So could it be that this high level of variation in the PRND gene is part of what's making cats more vulnerable to FSE?
That's a possibility. These variations might be creating a sort of genetic predisposition to prion diseases in cats.
This is all very compelling, but I imagine there's still much more to learn about the intricate relationship between these genes and FSE.
Absolutely. This research is a very promising start.
Yeah.
But we're still in the early stages. More research is needed to confirm the direct involvement of these variations in FSE and to fully understand how they influence disease susceptibility.
It seems like there's a whole new avenue of research opening up. What are some directions future research could take to build on these findings?
That's the crux of the matter.
Okay.
We'll delve into that and more when we return for part 3 of our deep dive.
Welcome back to the final part of our deep dive into the world of feline PRND and its potential role in FSE.
Yeah, we've covered a lot of ground.
We have.
From, a prion disease refresher to those newly discovered PRND variations in cats.
Right.
And their possible connection to FSE susceptibility.
And that intriguing link between the PRND variations and the indel in the PRNP gene.
Right.
The one that might influence how fast the disease progresses.
It's like we're putting together pieces of a complex puzzle, and we're starting to see a picture emerge.
Exactly. So let's talk about the future. Where do we go from here?
Yeah.
What kind of research could be done to build on these findings and give us an even clearer picture of why cats might be more susceptible to FSE.
Well, one exciting avenue is to go beyond computer analysis and actually look at whether these variations in the PRND gene lead to differences in how the protein functions. We can do this in the lab by producing the different versions of the protein, each with its specific variation, and then observing how they behave.
So we're talking about taking those genetic variations, those typos in the code, and actually seeing how they change the final product, the protein itself.
Precisely, we can see if these variations affect the protein's shape, its stability, its ability to interact with other molecules, all those factors that could influence its role in the cell.
That would give us real tangible evidence about how these variations might be contributing to FSC susceptibility. What else could be done?
Another important step is to expand the study population. This research looked at 210 domestic cats, which is a great start. But imagine the power of looking at thousands of cats from different breeds and different parts of the world.
So casting a wider net to see if these variations are common across different cat populations.
Exactly.
And if they're associated with different levels of FSE risk.
Exactly. We could even look at wild feline species and see if they share these variations and how that relates to their susceptibility to prion diseases.
That would give us a much more complete understanding of how these variations are distributed in the feline world. And what role they might be playing in prion diseases.
It would, and here's where it gets really interesting. Imagine if we could use this research to develop a way to identify cats that are at a higher risk of developing FSE.
So a genetic test for FSE susceptibility.
Yeah.
That would be incredible.
It would allow us to focus preventative measures on those cats that are most at risk. And potentially even develop targeted therapies in the future.
That would be a game changer for feline health.
It would. This research, while still in its early stages, has opened up so many possibilities. We've gone from knowing very little about the role of PRND in cats to having a whole list of new genetic variations that could be key players in FSE susceptibility.
It's amazing how a single research study can ignite so much curiosity and lead to so many potential avenues for exploration.
This is a testament to the power of scientific inquiry and the potential for groundbreaking discoveries that can improve the lives of animals and humans alike.
This has been a fascinating deep dive into the world of feline genetics in prion diseases. Thank you for guiding us through it.
It's been my pleasure. This is cutting-edge research.
It is.
And it's exciting to share it and discuss its potential impact on feline health.
From Vet Neurojar, keep those minds-inspired hearts light and tails wagging.